Next coming to the topic is hemopoiesis. Hemopoiesis is a process which involves origin, development and maturation of erythrocytes, leukocytes and platelets. Okay, so how the blood cells are developing or and maturing, so that is called hemopoiesis. Okay, so coming to the next one is the erythrocyte. We will study the RBC, what is its structure, what is the function and all. So it is one of the formed elements of the blood. It is circular, biconcave and non-nucleated disc. It is a circular structure. Okay, so when you see in a 2D direction, if you see it looks circular. It is a biconcave, so that means biconcave. So in the middle it is sides are bulging, so it is the middle, you can see here. So sides are not bulging. Okay, so in the middle it is bulging, so it is like this. Okay, so biconcave structure. So biconcave structure. Non-nucleated, the nucleus is not present. Okay, if you see, all the cells in the body is having a nucleus. So nucleus mainly contain what? The chromosomes or a genetic material. But the RBC cells do not have nucleus. Okay, then the diameter is 7.2 micron, so that is very small, very small. It's a microscopic structure. So then surface area is 135 square micron and volume is 80 to 94 cubic micron. Okay, so this is the structure, how it looks under microscope. Okay, so it is as I told, it's a biconcave structure. So what is the use? Okay, so what is the use of biconcave structure? So first one is it helps in equal and rapid diffusion of oxygen. So the main function of RBC is only to carry the oxygen and carbon dioxide, right? So easily, because of this biconcave structure, so easily the cells oxygen can be diffused into the blood. Okay, then it has a large surface area, so that will provide for absorption and removal of different substances, like removal of carbon dioxide and absorption of oxygen. It minimum tensions will be created on the membrane when the volume of the cell alters. That means if the cell structure or the volume, if it is any alteration in the volume of the cells, so it will provide a minimum tension. Otherwise what will happen, if the tension is more on the cell, so the circulation of the blood will be increases, so the blood pressure will be increases. Okay, it will provide a minimum tension if any alteration in the volume of the blood. So another one is while passing through the capillaries. So capillaries are very thin, very small. Okay, so while passing through the capillaries, so these cells can easily squeeze through the capillaries. Okay, so easily it can squeeze. Okay, squeeze means coming outside. Okay, so while passing through the capillaries, easily it can squeeze through these capillaries. So these are the advantage of biconcave shape. So this is the structure. For functions of the blood, so mainly there are three functions. One, the transport of oxygen. Okay, so mainly the oxygen and carbon dioxide. So oxygen from lungs to tissue and carbon dioxide is from tissue to lungs. Okay, so another one is because of the presence of plasma proteins in the blood. So RBC, because of presence of hemoglobin in the blood, in the RBC. So it is mainly regulate the acid base in the body, okay, or regulation of pH of the blood. So very important. So everyone will ask what is the functions of RBC. You should know mainly it transport the oxygen and carbon dioxide from lungs to tissue, tissue to lungs, then maintain the acid base in the body because of presence of hemoglobin. Okay, next is the erythropoiesis. Erythropoiesis is a process which involves origin, development and maturation of erythrocyte. That means how the erythrocytes are generating, so where it is forming, okay, like that. So origin, where it is origin, developing, then maturation. After the formation of the cell, the maturation is very important, okay. So to perform their function, maturation is important, okay. So that is called erythropoiesis, process which involves origin, development and maturation of erythrocytes. So next comes the site of erythropoiesis, where it is taking place in the fetal life, okay. So the origin is different in different lives, okay. That means in a fetal life it is different, in newborn babies, in children, in adults, okay. So in the fetal life, so it will form in 3 stages, okay. So it will form in 3 stages. One is the megaloblastic stage, sorry, mesoblastic stage, hepatic stage and myeloid stage. Understood? Erythropoiesis is the formation of RBC cells. So this formation is taking place in fetal life. It is 3 stages, okay. Mesoblastic stage, hepatic stage and myeloid stage. Mesoblastic stage, so that is between 3rd week to 3rd month of the fetal life. Understood? And it is in the uterus, when the babies are developing the uterus, that is the fetal life. So 3rd week to 3rd month, it is the RBCs are producing in the mesenchyme of yolk sac, okay, mesenchyme of yolk sac. Then hepatic stage, so it is coming developing from liver, spleen and lymph nodes. Then at the end, the myeloid stage, that is after 8 months, so it is developing from red bone marrow. So now after birth, okay, in postnatal life, after birth, the newborns, all the bones with red bone marrow, okay. So mainly the red bone marrow is very important, okay. All bones with red bone marrow, then liver and spleen. In children it is red bone marrow, and in adults it is 18 to 20 years, mainly from the red bone marrow of skull, vertebra, ribs, sternum and pelvis, okay. So this is, so that means the mainly it is forming from the bone, okay. So in the bone there is a red bone marrow is there, okay. So this is the marrow cavity, so this is marrow. So from the marrow the red blood cells and other WBC cells, so everything, okay, everything is developing from the red bone marrow. Understood? So erythropoiesis is the process of origin, development and maturation of RBC cell, red blood cells. So mainly this is formed in the fetal life. It is, there are 3 stages, and in the adults mainly from the red bone marrow. Next comes stages of erythropoiesis. So this formation of RBC is taking place in different stages. Okay, so suddenly it will not form one big cell, suddenly it will not form. Okay, so there are different stages are there. Okay, so you have to, very important. Okay, this question is very important. The erythropoiesis, how the cells are forming. So you can see the diagram. So for everything there is a stem cells. Okay, the stem cells are there. So this is the stem cell. So that is called pluripotent stem cell. Okay, so pluripotent stem cell, this is pluripotent stem cell. This is committed cell. So from the stem cell, so pluripotent stem cell is like a mother. Okay, so from the pluripotent stem cell, PPSC, pluripotent stem cell, there is a, it will form a committed cells, and we call it a proerythroblast. Okay, we call it as a proerythroblast. So from the proerythroblast, it is forms a normoblast. So normoblast will form a early, intermediate and late normoblast, then reticulocyte, then RBC cells. So in this picture, so what you can observe is, so here the nucleus is present. Okay, this is nucleus. Nucleus is present. Slowly the nucleus becomes small and once it will disappear. So in this reticulocyte there is no nucleus, right? No nucleus only. That is one thing. And the size of the cell. Okay, so this is bigger. The first one is bigger. Then the committed stem cell, the size will be in decreasing and it will form, the matured cell is will form a 7.2 micron. So that is the second changes. Okay, and the third changes is the color. Okay, so if you stain the RBC cells, can easily make out the changes, color changes. Okay, so the first one you can see the blue color, bluish. So then it is go on, so changing the color and become the red cells. That is for the accumulation of hemoglobin. So I will explain the each step. So understood, the first, the erythrocytes is developing from the pluripotent stem cell. The pluripotent stem cell will form a committed stem cell. So that is a proerythroblast, then early normoblast, then intermediate normoblast, late normoblast, reticulocyte and mature RBC cells. So understood. So erythropoiesis is the process of origin, development and maturation of erythrocytes. So this mainly origin is take place in the bone marrow. So that is different. So in fetal life it is, there are three stages: hepatic stage, mesodermal stage, okay, and myeloid stage. In the mesoderm of yolk sac mainly produce the RBCs in the first 3 months of fetus. Then liver and spleen in the next 3 to 6 months in the hepatic stage, and from the red bone marrow the mainly, so that is myeloid stage. So in the newborn babies, spleen, liver and spleen and also bone marrow in children and in adults, mainly from the red bone marrow. Okay, red bone marrow. So coming to the stages, you can see when you stain the cells, so you can see the different color changes and also the size of the cell will be changing. So it is initially it is bigger with nucleus, and slowly, so when it is coming to the different normoblastic stages, the size will be decreasing and color will be changes, and it attain the RBC color, that is red color, that is mainly by the presence of hemoglobin. Okay, the presence of hemoglobin. So next is the proerythroblast. So in the proerythroblast, the cell size is 20 micrometer. So that is very big. It is having nuclei because the nucleus is present and there is no hemoglobin. Then it is a basophilic stain, the basophilic cytoplasm are a blue color. The cell shows active mitosis. Okay, so mitosis mainly takes place in the nucleus, chromosomes, right? So mainly in the cell the nucleus is present, so mitosis is taking place, cell division is taking place. Okay, that is in proerythroblast. This is the future of the proerythroblastic stage cell. Now, so from the red bone marrow, one of the pluripotent stem cell will form a committed stem cell. So this committed cell form a proerythroblast. So this cell is having the size of 20 micron, having nucleoli and nucleus. There is no hemoglobin. Basophilic stain. Okay, there is acidic stain and basophilic stain is there. So basophilic stain, it appear, then it show the active mitosis, that is proerythroblast. So next, from the proerythroblast, it is going to form a early normoblast. Okay, it is forming a early normoblast. The early normoblast having a size is 14 to 16. Okay, so you can see that is 20. This is 14 to 16. That means it is decreasing. Okay, 14 to 16. There is no nuclei here, but has only the nucleus is present. The nucleus size also is decreasing. Okay, so not only the cell size, the nucleus size is also decreasing. No hemoglobin. If you stain, it show the basic stain and it show the mitotic division. Okay, the mitosis is present, or it show the active mitotic division in the early normoblast. That is the second stage. Next, coming to the intermediate normoblast. Intermediate, so that is in the middle. Okay, so in the intermediate normoblast, again the cell size decreases about 10 to 14 micron. Okay, 10 to 14 micron. Still the nucleus is present, but size is decreasing. Okay, so in the nucleus, the chromatin undergoes a further condensation. No? Further condenses. Okay, so next the hemoglobin starts appearing. So they will ask the question, in which stage of erythropoiesis hemoglobin starts appearing? About these two stages, early normoblast, pronormoblast, there is no hemoglobin. So in the intermediate normoblast, the hemoglobin starts appearing and it shows the mitotic cell division because the nucleus is present, mitotic division is taking place. That is in intermediate normoblast. So you can see the changes. In the first stage, the cell size is more and the nucleolus is present. Size is also big. Coming to the early normoblast, the cell size decreases, the nucleus size decreases, the nucleolus disappear. Coming to the next stage, the intermediate normoblast, the cell size still decreases because it has to come to the 7 micron. The RBC size is how much? Diameter is 7.2 micron, right? So it has to come to that level. Okay. So then only the maturation is taking place. So next coming to late normoblast. So this is here the cell size again decreases to 18 to 20. The nucleus become very small. Okay, so this is the nucleus. So nucleus become very small and attain a pycnotic. Okay, become pycnotic, then undergoes lysis. Understood? Will disappear. The nucleus become pycnotic and undergoes lysis and disappear. The hemoglobin content increases. Okay, the cell color you can see it become acidic or eosinophilic color, red color. So here because the nucleus is not present, the cell division will stops. Okay, the cell division stops. That is in late normoblast. So next come to reticulocyte. That is the next stage, just prior to maturation. So here the cell size is 9 micrometer. Okay, cell size is 9 micron. So here little bit remnant of RNA is present. Okay, remnant of RNA appears in the reticulum of cytoplasm. Hence it is called reticulocyte. Here there is no nucleus. You can see the remnant of. So these are the remnants of RNA. So no nucleus. So no cell division. Hemoglobin content increases. So the cytoplasm is eosinophilic. It is more red stain it is taking place. So next the last one is mature erythrocyte. So after reticulocyte, then the cell size further increases and it becomes 7.2 micron. So there is a lot of hemoglobin, no nucleus, no cell division. Cytoplasm staining is eosinophilic, present both in bone marrow and peripheral blood. Means mature erythrocyte is present in peripheral blood. Okay, if you see in the microscope, in the peripheral blood if you take, so that is mainly contain the mature RBC. So whatever you are seeing the reticulocyte or a late normoblast, intermediate normoblast, so these cells are not present in the peripheral blood. Peripheral means circulating blood. So mainly it is performing the function. The duration of erythropoiesis. So mainly, so from the pro-erythroblast stage to become reticulocyte, it will take 5 days. Okay, it will take 5 days. To become mature RBC cells, it takes 2 days. So total 7 days. Okay, total 7 days it will take. Understood? The red blood cells are formed in the red bone marrow. So from the red bone marrow, one of the stem cell become pluripotent stem cell, then committed stem cell. This committed stem cell we call the proerythroblast. So from the proerythroblast, there is a different early normoblast, intermediate, late normoblast, then become reticulocyte. So it will take 5 days to the cells to cross. Then from the reticulocyte to become a mature RBC, it takes 2 days. So total erythropoiesis. This is I am talking about the one cell. Okay. So one RBC cells. So like this, there are millions of RBC cells. So totally in the blood, normal count 5 millions per cubic millimeter of blood in adult male. So if you check the adult male, the normal RBC cells count is RBC count is 5 millions. Okay, and in female it is 4 millions. It is less than the male. Okay, in female it is only 4 millions, and in a newborn and infants it is higher, 7 to 8 millions. Okay. So I will stop here only.